Thank you, Mr. Birana. Good afternoon and welcome to the Institute Colloquium today. Today the speaker is Professor Narendra M. Dixit. He is the chair of Center for Biosystem Science and Engineering. He is also professor in the Department of Chemical Engineering. A brief introduction about the speaker. Narendra has a BTech from IIT Bombay and MS and PhD from University of Illinois at Urbana-Champaign, all in chemical engineering. He did postdoctoral work in theoretical biology and biophysics at the Los Alamos National Lab. He is currently Professor of Chemical Engineering and Chair of Center for Biosystem Science and Engineering at IISc, and is a Senior Fellow of DBT Wellcome Trust India Alliance. Today he is going to speak on a very contemporary topic. It's titled as From the Specific to the General and Back, HIV-AIDS, COVID-19 and beyond. Over to you, Narendra.
Thank you very much, Professor Navkanbhat, for those very kind words. It's a great honor for me to be delivering the Institute colloquium. I must also thank Professor Bhatt for the opportunity to represent the Division of Interdisciplinary Sciences here. It's a privilege. So as Professor Bhatt said, the talk is about viruses, but it's also about the nature of the interdisciplinary work that goes on at the interface of the physical sciences and biology. This audience needs no introduction to the nature of the work in these two disciplines per se. Physical sciences look for general principles. Biological sciences look for exceptions for rare phenomena. And in our work, we have had the opportunity, because we straddle both these disciplines, to abstract general phenomena from seemingly diverse biological observations, and then to tailor these general phenomena back to specific situations so that desired outcomes can be engineered. So I hope that through this talk, you will see this spirit working at many places. So let's just get started with the colloquium. Here's the outline of the presentation for today. I'll start with a one-slide introduction to our work, an overview of our work, and then step into the main problem that I'd like to look at, which is to think about the outcomes of viral infections from a dynamical systems point of view. Then I'll look at the implications of this view for interventions, and then I'll address two specific problems or cases. One is to look at HIV and this concept of functional cure, which I will describe as we go along. And then, as Professor Bhatt said, something even more contemporary, which is COVID-19 vaccinations. So let's get started. So here's a one-slide overview of our work. We're focused on infectious diseases, HIV, hepatitis C, TB, and more recently, SARS-CoV-2. And we've looked at these infectious diseases mostly from a computational perspective, but at many length and time scales. So we've thought about processes at the intracellular level, because viruses get inside cells and hijack their replication machineries, make more copies of themselves and spread. And to think about these processes, we use ideas from reaction networks, dynamical systems, and stochastic simulations. We also looked at how these infections spread within the body and how interventions curtail these spread. And there we use ideas from population and dynamics, spatiotemporal analysis, and also drug pharmacokinetics. And in response to these interventions, usually pathogens evolve, and our immune system also evolves in response to the evolution of the pathogens. So we study those processes using ideas from population genetics and evolutionary theory, and of course immunology. And lastly, of course, we have looked in some cases of the spread of the diseases in populations, looking at ideas from epidemiology. And here again, we look at spatiotemporal analysis, intervention strategies, and these ideas called nested models, where based on interventions, these pathogens evolve, and that evolution in turn influences the way they affect cellular processes. And this cycle sort of continues. Turns out we have not looked at the entire cycle for any one specific problem, but have looked at many of these linked processes for several examples. Today, I'm mostly going to talk about the bottom left two processes, and to some extent this, and in the context of HIV and hepatitis C and viral infections in general. So let me get started with the first problem that I want to look at, which is to think about the outcomes of viral infections. And this, as we all recognize, is extremely current. We have all been hearing of terms like asymptomatic infection, severe infection, mortality, and so on. So where does this all come from? I might say the virologists have been bothered by this difference in the outcomes of viral infections for a long time. So they have thought about this in the following way. Studies have been done using mice, usually, where infections of viruses have been introduced and viral titers have been introduced and the outcomes have been examined. Typically, virologists think of these outcomes as belonging to three categories. There is clearance, immunopathology, and persistence, and I'll describe what that is. Here is an example of one such experiment where these mice were infected with what is called lymphocytic chorea-meningitis virus, LCMV for short. What is shown here is the time course of the concentration of this virus in the spleen of these infected mice. And if a low amount of the virus was used for this inoculation, then the viral levels rose, came down, and then went away. And the mice were free of this infection. This is a histology of the spleen tissue, and it shows more or less normal tissue at the end of this infection period. If intermediate levels of the virus were used, then again you saw this rise. But then, as the viral infections declined, you started seeing large-scale death of these mice, and the tissues in the spleen were severely scarred and damaged. This is what we call death because of immunopathology. The last is persistence, where here the viral levels rose, came down, but then never vanished, and instead settled to some low level. And here the tissues were almost as normal as they would be in the uninfected case, but the virus was still there, it wasn't gone. So here you now had long-term persistence of this particular infection. Now, different viruses have different percentages of these very three outcomes that are realized in real life. So let me summarize those outcomes for us. HIV infection invariably causes persistence. There is no case of spontaneous clearance of HIV so far. Measles, 100% clearance, no persistence. Hepatitis C, 25% clearance, 75% of the cases you get persistent infection. Influenza, you get X% clearance, and the remaining actually pass away because of immunopathology. There is no persistent infection, and this X depends on the strain. So, for example, whether you have an H1N1 or an H5N1 and so on, and that will determine this X. What happens with hepatitis B? It's persistent in children, in neonates, but it's spontaneously cleared in adults. With LCMV infection in mice, which I just showed you, the outcomes depend on the strains of the virus used. There's something called an Armstrong strain, which is invariably cleared. And then there's something called a clone 13 strain, where the outcomes are dependent on the inoculum size, as I just showed you in the previous slide. And then, of course, the most recent SARS-CoV-2 infection. More than 95% of the people in general get rid of this infection spontaneously. They may suffer varying degrees of severity of the infection, but they finally are free of the infection. And of course, some percentage of the people unfortunately die. There's been no case reported so far of persistent infection. So what determines these outcomes, and can they be controlled? So this was the question. And this also has a long history of investigation in virology. I'll give you a brief history of this investigation, again in one slide. There were many factors that were identified as being responsible for these outcomes. And the way these factors are identified is by altering those factors and then examining whether the outcomes are changed. If there is a correlation, then that factor has been identified as being attributable in terms of the outcomes of the infections. So the first one identified was viral genetics. So if you mutated the viral genome, then you found the outcomes were different. The viral inoculum size, as I just showed you. There's this effector CD8 T cell response. This is an important arm of our immune system. These are cells that kill infected cells, virus infected cells, and this will revert to a lot going forward. This is an important player in today's talk as well. These were responsible for the outcomes. Natural killer cells, another arm of our immune system, they were also found to be responsible. Innate immune responses, many of you might have heard of this now in the context of SARS-CoV-2 infection. For example, interferons, which are chemicals that our body secretes in response to these infections, and they are responsible for outcomes is what has been inferred. Host genetic factors, if you are predisposed to a certain outcome, then of course you tend to suffer that outcome more than others. And this is even more apparent today with all the comorbidities that people are seeing in the context of SARS-CoV-2. And so on. And this list doesn't seem to end. There's a long list here. So if you read the literature, it appears that the outcome is the result of a complex interplay of a large number of factors. And as recently as 2018, papers were being written titled like this: LCMV clone 13 infection causes either persistence or acute death dependent on interferon 1, cytotoxic T lymphocytes, which are the CD8 T cells, and host genetics. So playing up this business of the whole scenario being extremely complicated. Our thought was, while this may be true, the outcomes are in the end just three. You either clear the infection, you become persistently infected, or you suffer immunopathology related mortality. So there must be something central to this whole process. And if it is the case, if we can identify it, then there might be a way for us to deal with these infections in a far more systematic manner. So that was our attempt, and we looked at the literature more closely. And we found that the interactions of these CD8 T cells and antigen, and in this particular case, infected cells, were critical to determining these three outcomes. So I'll give you now the outcomes of our study and why we feel that this is really the case. There are studies that say that CD8 T cells are absolutely necessary for clearing the virus. If you don't have CD8 T cells, you almost can never get rid of the infection completely. Persistent infections are associated with dysfunctional CD8 T cells. So if you don't have functioning CD8 T cells, then you have, this is more or less the same statement in another way. But the introduction here is of this notion of dysfunctional CD8 T cells. Antigen stimulates CD8 T cells. So how are these CD8 T cells even mounting a response? They get stimulated by antigen, and in this particular case, often infected cells. And the CD8 T cells, in turn, they destroy these destroy the antigen, or clear the infected cells. At the same time, sustained or strong antigenic stimulation of these CD8 T cells causes these cells to become what is called exhausted. So they now are no longer able to perform their functions with the same efficiency as they were doing otherwise. Now, this exhaustion has been argued to be a design principle to prevent immunopathology. The thought here is, if you had strong stimulation, it means that the pathogen is intense, it's more virulent, and spreading rapidly. And at the same time, if you have an aggressive CD8 T cell response, then it can destroy a lot of tissue and produce immunopathology. And it's to prevent that evolutionarily, this notion of exhaustion has gotten selected. And this is therefore thought of as this design principle to prevent immunopathology. Reversal of exhaustion can prevent persistent infections. I can actually clear persistent infections. So if you had a sustained infection and you somehow manage to reverse this exhaustion, then you can actually be free of the infection. And this, of course, has been demonstrated in a tremendously powerful way for cancers, and it won the 2018 Nobel Prize. So if you think about this literature, you find that in the end, the interactions of these CD8 T cells and antigen that appear necessary and sufficient to determine all the three outcomes that we examined earlier. So based on this premise, we constructed the following hypothesis and said, let's look at whether this makes any sense. So our hypothesis was that there's a dynamical motif that comprises the essential interactions of CD8 T cells and antigen, and this defines the outcomes. And all the other factors that have been listed perhaps work by modulating the interactions in this particular dynamical motif, and this therefore influence these outcomes only indirectly. So is this even right? How do we test this? So we set up this dynamical motif and looked at what its implications are. So this is the motif. It's extremely simple, and the analysis that follows will also be simple, but then it has the necessary ingredients for us to draw the inferences that we would like to. The two players are the infected cells. This is the antigen, and these are the CD8 positive T cells or effector cells. What are the interactions? Infected cells, of course, they grow because they produce viruses, and those viruses go and infect other cells, and therefore this population keeps increasing if there was nothing else going on. These infected cells, of course, interact with these CD8 T cells directly and indirectly, and they activate these effector cells. These effector cells can in turn kill these infected cells. And if you have a lot of these infected cells, then these would cause these effector cells to become exhausted. Now these stubs here indicate suppression and the arrows indicate activation. So how do you now analyze the outcomes of this particular motif? So we wrote down some very basic equations describing all of the interactions here. This is an extremely minimal model, and I will make the model more sophisticated as we go along. This is the population of infected cells, and this equation tells us how this number changes. If there's nothing else going on, this is called a logistic growth term. The number rises and then it saturates at a maximum value determined by the resource limitations in the system. If there are effector cells, they destroy these infected cells, and therefore their number decreases in some mass action manner. effector cells get stimulated by infected cells in a saturable manner, and they also cause exhaustion of these very cells. Thanks. Yeah. They cause exhaustion of these very cells, again in a saturable manner. And these parameters K3, Kp, K4, and Ke are adjusted in a way that when the infected cell populations are small, this term dominates, and when they are large, then this term dominates. So initially you'll have stimulation and then you'll have exhaustion. Now these are standard terms used in the literature, and so we have sort of borrowed those terms to set this model up. So what happens when this model plays out and reaches some kind of a steady state at long times? So we look at the stationary states of the system, and you find that there are three stationary states. One is where there is no infection and you are left with a certain population of effector cells, and this state, if you do linear stability analysis, will find is stable. The other state is that you have a large population of infected cells and no effector cells. Of course, these are limiting cases, and this state is stable as well. And then there is a third state where both infected cells and effector cells are non-zero, and this is actually an unstable state, but it's a saddle. So these are the three states. Now, which states you realize depends on initial conditions, like in any other dynamical system. So to understand that, we looked at a phase plane plot. So here's the dynamical system. Here's the phase plane plot of this dynamical system. X-axis are infected cells, y-axis are these effector cells. The solid red circles are the stable steady states. This is where you have very high infected cell populations and almost no effector cells, and this is the persistent state. This is the clearance state where you have very low infected cells and very high effector populations. And this open circle here is the unstable saddle. Now here you get attraction to this steady state in one direction and repulsion in another, as in other dynamical systems. Now there's a red line here that passes through this unstable boundary, unstable steady state, and this separates this phase space into two parts. One where if you start this infection in this region, you would end up in this steady state. And if you start the infection in this region, you would end up in the other steady state. So this then defines the way in which you reach these two different outcomes. So now we look at these individual trajectory, these lines here indicate trajectories. So if you start out here, and these arrows indicate the progression of infection. If you start out here, you probably go like this. Your infected cell population and effector cell populations would increase, and then you would eventually hit persistence. If you start out here, again, you would go like this, and then you would eventually hit clearance. So if you look at these as time courses, you will find that playing out. If you're on this side of the phase boundary, you would have infected cells reaching higher values. And if you're on this side of the boundary, you would have infected cells reaching low values. The opposite happens with the effector populations. Now, so this basically tells us that these clearance and persistence look like steady states of this particular system. Now, what about immunopathology? That turns out is a dynamical outcome. And how do you understand that? You look at what pathology is. It's really proportional to immune killing of infected cells. So if I think about the extent of pathology as arising from the extent of killing of infected cells, which is basically this mass action term, and then there's some decrease in pathology because of a natural clearance of the cytokines that are released in the process, then one can estimate the extent of pathology that one experiences. And you find that the pathology is most when you're very close to this phase boundary. So why does that happen? If you're on an extreme on this side, where you're supposed to hit persistence, what happens here is you have a lot of infected cells relative to these effector cells. So you quickly cause exhaustion of these infected cells, and therefore not enough tissue damage happens. You go to the persistent state. If you're far into this region here, then you have a lot of effector cells and very few infected cells, and those infected cells get cleared quickly without causing too much tissue damage. So again, you don't get enough pathology. But if you're close to this boundary, then because of the nature of the phase space, you execute large trajectories along this boundary, come close to the saddle point, and then deviate. In the process, what happens is you build up a lot of infected cells and effector cells. And why is this happening? You have a lot of infected cells, they trigger effector cells, but you don't have enough infected cells for triggering exhaustion. So both these populations rise, and in the process, you cause a lot of tissue damage. And that's how you get this huge pathology. So this pathology is really a dynamical outcome of this particular motif, if this were to be believed. So does this really play out in experiments? So we looked at this from some experiments that were reported in the literature. This is again LCMV infection of mice. These were infections, these were experiments where low, medium, and high viral loads were introduced in these mice. And what's shown here on the left y-axis is the percentage of the mice that were persistently infected. And on the right y-axis in red is the percentage of the mice that died because of pathology. So what you find here is if you have low viral inoculum, nobody dies, nobody is persistently infected. Everybody clears the infection. If you have very high virus, again, nobody dies, but everybody has persistent infection. And at intermediate viral levels, you start seeing death. This is even more evident when you look at these experiments where CD8 T cells were introduced artificially into these mice. And then the mice were subjected to this infection. And low, medium, or high levels of these CD8 T cells were introduced. And when that happened, here's what happened. When you had low CD8 T cells, nobody died, everybody was persistently infected. When you had high CD8 positive T cells, everybody cleared the infection, nobody died. Medium CD8 T cells, in this particular experiment, everybody died. And so this was clear that you had a lot of pathology when you are intermediate levels. So this is basically thought of from this phase diagram in the following way. This experiment is basically cutting across the phase boundary laterally. You increase the viral levels. So as you go up like this, you first clear, then suffer pathology, and then become persistently infected. And you can see that playing out in our calculations: clearance, persistence, peak pathology in the middle. And this experiment, you're cutting across the phase boundary like this, from the bottom to the above. And here, again, persistence, pathology, clearance, persistence, pathology, and clearance. So this is basically telling us that this particular set of observations is correctly captured by this view of a dynamical motif of this infection. So what happens to all the other factors? So let's take one example. Innate immune responses. This is important because of all the virus SARS-CoV-2 literature going around. What do innate immune responses do? This is now a lumped version of the innate immune responses. Everything associated with innate immune responses is here. They're triggered by infected cells, and what they do is they prevent viral replication. So they basically reduce this growth of these infected cells. That's the basic way of thinking about what innate immune responses do. So if you do this now, what happens? You can redo the analysis. Of course, now the system has grown in dimensions. It's a 3D system now, so visualization is very difficult. The phase boundary now becomes a surface. But then just to aid visualization, I'm going to look at a projection. The way to think about this now is that the separatrix of the phase boundary has basically been moved because of the innate immune response. How? If you have a strong innate immune response, it suppresses the growth of these infected cells. So a lower amount of the effector cells are now able to bring about clearance. So this phase boundary basically shifts below. So this point, which earlier would have gone off to persistence, now gets cleared because the phase boundary has moved around. So this is basically telling us that these innate immune responses work by altering some of these interactions here. Now, is this played out in experiments? So we looked at some experiments of hepatitis C virus infection of monkeys. So what happened here? Same virus, same inoculum, same strain of the virus, and everything controlled. These monkeys were infected, and the viral levels as a function of time were measured. This is the viral level in the blood. Again, you found two outcomes: clearance and persistence. So the viral levels rose. I'm going to focus on these square symbols here. Then the rise slowed down, and then it came down. And in this particular case, it was completely eliminated. Here too, the same thing happened, but then here the virus went on to become persistently infected. So now to understand this, we set up the same sort of description as earlier, but then we expanded the model to incorporate the production of the viruses explicitly, and then some details about hepatitis C virus infection, where the target cells proliferate, they get infected by the virus, and then they become infected cells, and these proliferate as well, because that's what liver cells do. Then we know all the parameters associated with these infections. This innate immune response is primarily interferon responses in hepatitis C infection. So we know something about these responses as well. So we can put some of these numbers into this system and ask what happens when we have low versus strong innate immune responses. And we managed to recapitulate those trends. You get the rise, and then the slower rise, and then a decline. In this case, followed by persistence, and in this case, followed by clearance. Here you had stronger innate immune responses. Here you had weaker innate immune responses. Now, and why does this thing work out? When you have strong innate immune responses, the viral levels are suppressed, the antigen levels are lower, they prevent as much exhaustion of the effector cells. So the effector cells are much higher, and they manage to clear the infection. So does this thing play out in those monkey experiments? So from the data, we looked at correlations of the following. the time when this transition happened from the sharp rise to the slow rise, that's the onset of the innate immune response, and then the peak of the viral load, which is a measure of the strength of the innate immune response. And we found that both these correlates were such that in the monkeys that had clearance, you found that the innate immune responses were triggered faster and they were stronger because they kept the peaks lower. So this basically suggested to us that this description of the innate immune response modulating the very essential interactions in the original dynamical motif were probably playing out. So they're curtailing viral loads and then allowing the CD8 T cell response to effect clearance. So we repeated this with NK cells with a whole bunch of other factors that have been identified earlier as being responsible, and invariably this description held out. So we thought therefore that this was a reasonable way of thinking about these viral infections. So this then has implications for interventions. So how do we think about interventions? Our goal in interventions would be to minimize pathology and effect clearance. How do you do that? You can do that by tuning each of these interactions. And I'll map now the various interventions that people have used in terms of how they influence these very interactions. So one way to effect clearance, for example, would be to reduce the number of infected cells that we have. And that can be brought about by drugs or therapeutic antibody vaccines, which is effectively like the plasma therapies of today. You could increase the CD8 T cells, and this is achieved using T cell vaccines. You could increase the activation of these CD8 T cells, and one way to do that is passive vaccination, and I'll talk about that in a bit in the coming slides. Exhaustion, and that's through immunotherapy, like checkpoint blockade inhibitors, which are used for cancers today also. And you could increase the killing of these infected cells by effector cells, and that is achieved using some developmental therapies called chimeric antigen receptor T cell therapies, and this is how these therapies are supposed to work. What each of these interventions would effectively do, if we were to assume this dynamical systems view, is cause a shift of the state of the dynamical system from it heading towards persistence of pathology to the region of the phase space, which takes you to clearance. So one other takeaway from all of this is that early interventions are far better than late interventions, because in early interventions, the effort you need to put in to cause this shift is much lesser. If you wait a long time, and if you've already come, for example, to somewhere here, then to cause this system to cut across the phase boundary is very difficult. So early interventions therefore help. So this is the way this dynamical systems view helps us rationalize current treatments. So now I'm going to look at examples of how some of these things are playing out in current treatments. I'll do that in the context of HIV infections. This is the second part of my talk. So, brief introduction to HIV infections. What is shown here is the concentration of HIV virus particles in the blood of an infected individual as a function of time following the contracting of this particular disease. So the virus initially rises, then the immune system kicks in, controls it, and then it settles, as I said, into a persistent infection. And this then lasts for many years. Notice the change in scale here. This whole dynamics was in weeks, and then the rest happens over years. But gradually here, the viral levels is rising. And at some point, the virus has caused the death of the target cells that it infects, the CD4 positive T cells, which are important parts of our immune system. And that death has caused such a severe hindrance to our immune system that the person then suffers severe immune compromised states and then dies because of AIDS. So this is the general progression of this disease. So to deal with this infection, people are put on antiretroviral treatments. So when one is put on antiretroviral treatments, the virus level quickly comes down. But then the moment you stop treatment, the virus rises again and then settles down to a level that almost was at the same level at which it was before the start of treatment. Because of this, one requires lifelong treatment. There has thus far been no case where the virus has been completely eliminated because of this virus. Invariably, the virus comes back. There are only few cases, in fact, I should point this out, where what is called sterilizing cure, where the virus has been completely eliminated from the body, has been realized. This is the famous case of what is called the Berlin patient, the Timothy Ray Brown. This person might even be the most famous case in recent medical history. It's a rather unfortunate case. This person suffered leukemia of the same cells that are targets of HIV, and unfortunately also contracted HIV. So he was being treated by Dr. Jero Hutter. And what Dr. Hutter realized is that to treat this person's cancers, cancer, he needed a bone marrow transplant. Dr. Hutter did something very smart. He found a donor for the transplant who had these target cells that did not have a particular receptor on their surface, a certain protein on their surface that the virus requires to infect them. Then he transplanted this person's bone marrow with those donor cells. Of course, this person was relieved from the cancer, and interestingly, he got rid of the virus. He got free of HIV. He was that way for many years, 10, 12 years. Is probably the first documented case where people think a sterilizing cure has been achieved. Unfortunately, last year he had a relapse of the cancer and passed away. But since 2 more patients have been argued to be cured that way, but this procedure is so complicated that it's not advisable to people who otherwise don't have complications like leukemias and so on. So the field has moved away from thinking about achieving a sterilizing cure of HIV and is thinking more along the lines of what is called as a functional cure. Can the virus levels be kept under check, under control, without having to worry about completely eliminating the virus, but then doing so in a way that does not require lifelong treatment? If we are able to do that, that state would be called functional cure. And the hope is that in that way, you don't damage the immune system to a point where AIDS develops. So can this be achieved? There's some hope that this can be achieved. Here's an experiment which gives us proof of principle. This is again done on macaques some years ago. Here these monkeys were infected with HIV, and then they were given treatments with antibodies, just like our plasma therapies. And the treatment was initiated extremely early, 3 days after the start of the infection. And the reason is, as I said earlier, at least as far as our understanding is, that you can effect changes much better if you do it early. So 3 doses of these antibodies were given. AB is short for antibody. N is neutralizing. So neutralizing would mean that these antibodies would prevent the virus from infecting cells. And B here is broadly. So this then works against the diverse strains of the virus. 3 doses were given, and then the monkeys were monitored for a long time. And here's what happened in many of these monkeys. The virus initially rose after the infection, but then was brought down because of these antibodies. But then when the antibodies were cleared from the system because of natural clearance mechanisms, the virus shot back up. But then interestingly, it was brought under control slowly, and then at some point it hit below detection levels and then stayed there forever. But the virus was still there in the monkeys. At this point, after many years, in this experiment, the CD8 T cells in these monkeys were depleted temporarily. And as soon as they did that, they saw a surge of the virus. And then once the depletion faded, the virus was brought again back into control. So this was suggesting that there was this possibility of achieving functional cure, where after these 3 doses of the infection, you had the virus completely under check. It wasn't gone, but it was at very low levels. It was not even detectable. And this was achieved in 10 out of these 13 monkeys. So how does this thing work? So we looked at our dynamical systems framework and asked if we could understand this. So here is a model of HIV infection following this dynamical systems framework. It's more or less the same model. You have the same interactions as I talked about earlier, except now you have target cells, which are the cells that the virus infects, and the virus explicitly described. And the other thing that is introduced here are something called antigen-presenting cells. And these are cells that take up infected cells and then stimulate effector cells. This is the correct process of effector cell stimulation. The reason you need to think about these processes explicitly will be clear in a minute. So if you now set up equations describing these players in this dynamical system, you find that this system actually admits 2 steady states, just like the previous one, but now the steady states are more nuanced. In one state, you have high virus, weak CTLs is CD8 T cells response, and high levels of exhaustion. The other state is low virus, strong CTL responses, and low levels of exhaustion. And what you find is invariably when you start the infection with a low amount of the virus, you hit this state. That's what the persistent state really is. This state is rarely realized. That's how the nature of the virus is. Now, what happens if you introduce antibodies? They do 2 things. They, of course, neutralize the virus, which means that they reduce the amount of virus that can actually infect cells. And the other is by coating infected cells and by viruses, they increase the amount of these viruses that are taken up by these antigen-presenting cells, and therefore increase the level of activation of these CD8 T cells. So these are the 2 dominant things that these antibodies do. There are other things that they do, but that we found were not critical to this particular process. So when we now set up this system, we found that these antibodies were now making this particular state much more accessible. they were driving the system dynamically to a region of the phase space where this steady state was becoming realized. Did this really work? So we were able to fit this model to actual data from monkeys, and quite a few of the monkeys were fit quite well. This was not easy to do, but was nice because it then told us that the model is reasonable. It's actually describing the underlying biology correctly. So if we look at one of these monkeys more carefully, you'll see what really is going on. So this is the dynamics. The blue line is our fit. The symbols are data from the monkeys. This is concentration of the virus. In the top panel, the CTL response and the level of exhaustion here. So the virus goes up, comes down, the antibodies are gone here. The virus comes back again, and then it's controlled. And the reason it's controlled is because here the antibodies have suppressed the viral load to a point where they have prevented sufficient CD8 T cell exhaustion, and because of the priming of the virus, they have left a lot more CD8 T cells stimulated by the time the virus comes up. So this now drives you to the region of the phase space where clearance becomes slightly, and in this particular case, that clearance is the low level steady state. It turns out the parameters are such. So then you hit this low level steady state, and therefore this is what is the realization of the other outcome through these therapies as we predicted. What happened if you did not introduce these antibodies at all? That's what is shown here in the red lines. You would hit the high infection steady state with everything else being constant, no changes in the parameters. That's where you would be. You would have a weak CTL response and a high level of exhaustion. So now that we know this, we can ask what amount of virus, what amount of antibodies can be used so that this cure or functional cure can be brought about in most monkeys. Now there were 10 out of 13, the others did not. So you can now tune the antibody concentrations and serum half-lives and identify regions of phase space where you would have progressive infection versus control. And you can show that if you were here, then you would progress. Whereas if you now administered antibodies at a higher concentration, you would control. Or if you had antibodies that are lasting longer in the body, then too you would control, because these would stimulate CD8 T cells much more. This of course is nice, but it works if you start treatment very early. Most people don't come to the clinic that early. So what happens if they come in late? If you come in late, and this has been tried with people, these are humans now who are being given these antibodies after their chronic infection. Unfortunately, it doesn't work. And our understanding is that it doesn't work because you're already in the state where the same concentrations that worked early in infection now no longer work. You need much higher levels of antibodies. And we are now making predictions of what those antibody levels are that would drive a system that is at this state all the way down to its low level control state. And that's for some other day. We haven't finished those calculations yet. But these studies where these patients were administered these antibodies late, they threw up another surprise. And I'll maybe talk about that very briefly in the interest of time, and then switch over to SARS-CoV-2. So what happened was that these antibodies, when they were administered in chronic infection, improved the ability of our body to produce antibodies, which was quite surprising. So this is how these antibodies were administered. This is one of the antibodies that was injected in, and after some time this antibody fades. Now in this experiment, antibodies from these people who were infected, who were injected this antibody, were drawn out from the blood, and their ability to neutralize HIV was measured before this antibody infusion and long after the infused antibodies were cleared from circulation. And what was the difference? Here was the interesting difference. If these antibodies were not administered, then there's almost no difference. So what is shown here is the difference in the ability of these antibodies to block virus infection versus these antibodies. Each bar here is an individual patient. So here you find that almost there's no difference if you did not administer these antibodies. But if you did, then you found that there was a huge improvement in these antibodies, in the ability of these antibodies to block viral infection compared to these. And this effect faded if the patients who were infused these antibodies were already on antiretroviral treatment and therefore had low levels of the virus. This is quite surprising because these antibodies are not supposed to trigger an improvement in the production of antibodies internally. So how does this thing happen? So we looked at this question in some detail. So this requires us to delve a little bit more into biology. You've got to think about how antibodies are produced, and they're produced in what is called the germinal center. The production process is called the germinal center reaction. Now these germinal centers are temporarily assembled sites of antibody production inside our lymph nodes. They have a specific and interesting architecture. There's something called a light zone and there's something called a dark zone. In the light zone are follicular dendritic cells. They have on their surfaces antibodies onto which are bound antigen. Now these are the ones that are going to trigger antibody production. They are also there B cells. These B cells are the ones that actually will produce these antibodies. They have on their surfaces B cell receptors. These are transmembrane versions of the antibodies. These antigens, suppose I now think about the antigen as a bit string of some kind, and these antibodies or these receptors as another set of bit strings. And I now think of the match or the ability of or the affinity of these B cell receptors for these antibodies. And I map that affinity onto the number of filled squares here. This is low affinity. This is almost no affinity. Low affinity, intermediate affinity, high affinity, and so on. What these B cells do is they snatch this antigen from these antibodies. When they do that, they get a survival signal. Otherwise, they just die. When they get a survival signal, they go to another set of cells inside these germinal centers called T follicular helper cells. And these T follicular helper cells give them a second survival signal. And when they receive that second survival signal, they can experience several outcomes. One is they exit this germinal center, mature into what are called plasma cells, and they become factories for the secretion of a large number of these antibodies. As I said, these are just non-transmembrane versions of these very receptors. The other fate that these cells experience is that they go back into the dark zone. And there they proliferate and they mutate their antibody, their receptor genes. And they therefore now have new cells with different kinds of genes, which may have lower or higher affinity for these very antigens. So these now come into the light zone again, they migrate, they re-enter, and then now there is competition for these antigens. The higher affinity B cells preferentially succeed in extracting these antigens. They get the survival signals from the T follicular helper cells. They then become plasma cells and now secrete antibodies that have higher affinity for these very antigens. And this is how you produce antibodies of increasing affinity for antigen. This process is called affinity maturation. This is how our immune system adapts, it learns. Some of these antibodies feed back into the germinal center and they then affect this process by taking on the role of the antibodies that are presenting antigen. And when you have external antibodies, they can do the same thing. They come into the germinal centers and they alter this antigen presentation process. And our hypothesis to explain the observations was the following, that if you have endogenous antibodies in the germinal center, that they would then steal these antibodies, antigen from the presenting antibodies and themselves assume the role of antigen presentation. And in that case, what happens is a B cell that should have acquired antigen from this receptor, from this antibody, now has to do it from this antibody. And if this antibody, which is administered externally, has much higher affinity for this antigen than the endogenous antibody, then this B cell has to do much more work. Only those B cells that have much higher affinities for this antigen than this antibody will now be able to extract B cells. So this is how these administered antibodies modulate the germinal center reaction and cause the preferential production of better antibodies. So we set up stochastic simulations of this whole process, and I'm going to skip some of the details in the interest of time. But what we were able to show is these are Wright-Fisher simulations which go through generation after generation of this affinity maturation process, mimicking the biology involved. And what we were able to show is that underlying this whole germinal center reaction is a quality quantity trade-off. So if you administer a lot of passive antibodies with high affinity, they increase the selection stringency inside the germinal center. That causes B cells that have much higher affinity for the antigen to be selected, but there are far fewer of those B cells. So you get a lower antibody output, although the quality of the output is much higher. Now this actually plays out quite well. If you look at a snapshot of this simulation, you find that as you go from low affinity passive immunization, this administration of antibodies is called passive immunization, intermediate versus high, you find that this the amount of cell death that you see inside the germinal center actually increases. The GC population size shrinks. the output is low, but the affinity of the output cells is much higher. And this is actually seen in mice exactly like this. So we therefore believe that this correctly describes the Germinal center reaction. Of course, the other thing that happens, and you can imagine that this might be the case, is if you have a lot more antigen inside the Germinal center, then the stringency of selection comes down because B cells have much more of a chance to acquire antigen. And this was seen in experiments where antigen was administered either in an increasing amount, decreasing amount, or a constant amount. And the increasing amount of antigen actually gave you a better response because it eased the selection stringency brought about because of antigen availability. And this again, our model was able to capture. And this gave us some confidence that this process was actually correct. So we now have a way of understanding this. This happens because the Germinal center reaction is modulated by these administered antibodies. And this happens because of the quality quantity trade-off. Here you have much less antigen because these people are already on antiretroviral treatment, and therefore the selection stringency is so large that although you get high quality antibodies, there are too few of them for you to see much of an improvement in the antibody response. So this, of course, now tells us that you can actually optimize now. If you have these antibodies at your disposal, how do you administer them so that you get the best response? And we did some of that optimization. I'm not going to go, I won't go into the details, but we have now the framework with which we can begin to think about passive immunization strategies as optimal ways of potentially bringing about functional cure. So this sets up the framework for us to think in the last 5 minutes or so about SARS-CoV-2 infection. So what do I want to talk about here? How do these vaccines work and why do we get the responses that we do? This is the response that is seen with the Moderna vaccine. What's the process? You give people 2 doses separated by some 28 days, wait for 14 days, and then you look at how many people who are vaccinated actually suffer the infection again. This is the percentage that suffer infection as a function of time if they were given placebo, and this is what happened when they were given vaccines. So this is what is giving you the 95% success rate. There's a 95% reduction in this number. So how does this work? It's not easy to understand what these vaccines are doing straight away. It's not like they produce antibodies. The problem is with the diversity of the antibody response. And I'll talk about that in a minute. So what are SARS-CoV-2 infections like if we think about this dynamical systems framework? 5% people suffer mortality because of immunopathology. 95% people clear the infection. Persistence is never realized. Out of these people, so most of the vaccines are basically working here. There might be some cases here, but that's so small in number that you don't see them in the results. Estimates are that 40% people are asymptomatic, 60% are symptomatic. So what the vaccines are doing is converting these symptomatic individuals into asymptomatic. That's the measure. And this is happening to different extents with different vaccines. The ones that we know, the mRNA vaccines have 90% efficacy, 90+% efficacy. A single dose Pfizer vaccine gives you 46%. And the Oxford one has variable numbers, and the most recent ones are 80. So how does this thing work? In terms of the dynamical systems framework, the vaccines produce antibodies that are now pre-existing. They would therefore drive the system deeper inside this clearance region of the phase space, and therefore effect clearance without much rise in the virus, and therefore you don't see symptoms. The problem is to describe what these antibodies are doing and how. So for that, I have to quickly describe to you how antibody responses are measured in people. So what is done is called a plasma dilution assay. So what you do is draw antibodies from people who have them. You use them in culture systems where you have viruses that are looking like SARS-CoV-2 viruses. They're supposed to infect cells. And now you look at this infection in the presence of the plasma drawn from these patients. So if the plasma were effective, very few cells would be infected. Now red are infected cells and blue are uninfected cells. But now you repeat this experiment by diluting the plasma. And as you dilute the plasma, fewer and fewer infections are prevented, and eventually you get a lot of infection. So you look at how this related infection proceeds as a function of plasma dilution. And the point where you get 50% reversal of infection is called NT50. This means that this is the amount of dilution that you need to achieve 50% of the block of infection that the plasma was originally able to do. So this is a good number. The larger this number, the better it is, because you have a much more powerful plasma. You need to dilute it much more for the infections to be reduced to 50%. The problem is, now this is what is measured, by the way, in patients who are given the vaccines. This is the Oxford vaccine data. you'll see that this dilution varies quite a bit over an order of magnitude. So some people have very low plasma dilution numbers, others have very high NT50s. And this is now convalescent patients who are patients who have recovered from the infection. And this number is also hugely varied. So how does this thing all add up, right? So this diversity of antibody responses is not easy to comprehend. So our thought was that based on the germinal center reaction that we just looked at, depending on various ways in which antigen is presented, stochastic effects, the kind of antigen that is looked at, the B cells that the individuals have in them, etc., one can imagine that there is a large pool of neutralizing antibody types that exists, and different individuals generate antibody responses that are drawn randomly from this pool in different amounts, in randomly drawn amounts as well. And this is in response both to the infection as well as to vaccination. So does this thing really work out? So how do you first of all identify this pool, right? So we looked at the effect of individual antibodies. So a lot of people have actually studied individual antibodies because of their intention to develop these antibodies for antibody therapies. So you again do these in vitro experiments where you have viruses exposed to cells and you look at how much infection happens, and you now repeat this in different concentrations of this antibody that you've isolated from people. So the infection, of course, this is the reduction in infection as a function of this concentration. So when this concentration is low, there's almost 100% infection. But as you increase this concentration, the infection is blocked fully. So this concentration at which this infection is blocked by 50% is called IC50. And the slope of this line is called M. And this data is very well described by this Hill function type expression. So if we know IC50 and M, we know what these antibodies would do at different concentrations inside these infected individuals, or in the way in which they would block infections. So just to make life simple, you can log transform this and you get a straight line. So it's a lot easier to analyze the data. So what we did was we looked at a large number of antibodies that are present, that are analyzed in the literature, and we applied these Hill function type equations and obtained the values of IC50 and M. So this is a snapshot of around 75 of these antibodies. And when we mapped it on this diagram of M versus IC50, they all lay in some region. This, for us, defined the landscape of the neutralizing antibody response to SARS-CoV-2. So different people would draw antibodies from this region. Now these colors are telling us how powerful they are. I won't go into those details, but as you go from here to here, you get more powerful antibodies. So now we looked at whether these random sampling from this landscape would describe for us what goes on with SARS-CoV-2. So we repeat the plasma dilution assays, but now instead of plasma from patients, we generate synthetic in silico plasma by sampling from this landscape. And what do you find? These are the plasma dilution curves. Each curve here is a different realization, so a different patient. And we can also now look at the concentrations of the antibodies in the plasma, and we can now compute NT50 as a function of the total antibody concentration in the plasma. The black lines are our calculations, and these symbols are data from a large number of studies. So this very simplistic view is actually describing this antibody response quite well. So the thought was that we could now use these to look at what happens in patients as a function of vaccination. So what happens with vaccination? You get again a sampling of antibodies from this landscape, but they now exist when the virus comes into the body. And now we go back to this model, looking at how the virus levels will rise and fall as a function of these antibody titers. So if I solve this model, here's what I find. Crudely, just when I keep the efficacy of these antibodies constant. If the antibodies are not there, the viral levels will rise and then they would eventually get cleared. This is the typical case. We are not looking at pathology and so on. As the efficacy of the antibodies rises, 99% of the infections are blocked here, for example, the peak of the viral level comes down. And this peak is basically a manifestation of the degree of symptoms. Eventually, the virus becomes undetectable and you see no symptoms. This is what happens. So now we can repeat this by drawing antibodies from our landscape explicitly and randomly and repeating these calculations. So when we do that, here's what we find. If the antibody levels are very low, this is the spread of the peak viral load that you would realize. It of course varies widely, and this is indicative of the diversity of the symptoms that people see. As you increase antibody titers, now these antibody titers are what are produced by the vaccines, you will find these numbers dropping. And the percentage that falls below this detection level is the percentage that now is protected by the vaccines. These are people who don't see symptoms. And you can keep doing this for increasing levels of the antibody titer, and eventually you get a large protection. So if I now look at the protection as a function of the antibody titer, I get this very nice sigmoidal curve. Now, how does this compare with the clinical data that people look at? This is the Pfizer data, NT50 as a function of time following 2 doses. So if I now, this is convalescent plasma data. This is 35 days after the first infection, the first shot. So if I look at the neutralization titers, I know the range of NT50s that are seen in these patients. From our plasma dilution assays, that range would correspond to this range of antibody titers. If I come to my protection predictions here, at that antibody titers, I'd get this level of protection. That's close to the number that is reported. I can do one more calculation. I can look at what happens if you just have a single dose at the end of 21 days. I can again come back and look at the IgGs that I should have. I come here at that IgG level, and we predict about 50% success rate. And this has just been published. If you just look at the single dose, the protection is 46%. So this is pretty damn good, in my opinion, in terms of understanding how these vaccines are really working. So with this formalism, we can now begin to ask how long will protection last? Because people can measure how these antibody titers wane. We can ask what happens if concentrations are changed and these antibody titers are altered, and so on. So we are at this stage right now. I'm going to stop here and thank people who have done all of the work. I put up photos of the people whose work has been described explicitly. Pranesh and Rajat were postdocs. Pranesh was also a PhD student with us. Subhashish and Rubesh were PhD students. Amar was an MTech research student. And for the outcomes of infection work, we also collaborated with a colleague, Rustam Antia from Emory University. I've collaborated with a lot of colleagues on campus and outside, and of course, a large number of students and postdocs working with whom I've learned a lot. So thanks to all of them, and thanks also, of course, to the funding sources. And thank you all for your attention. I'm happy to answer any questions if there are.
We can have the questions. You can post the questions on the chat box.
Sir, 6 questions are there in the chat box. 7 questions.
I can't see any in my chat box, Birana.
Sir, they are published, sir. You please go to the published, this one you can see.
I cannot see it.
No, I'm able to see some questions. Do you want me to
you
answer?
can. Okay. Yeah. Why don't you repeat the question and then answer? Yeah.
Answer. Okay, fine. So Nikhil asks, histology shows presence of nucleus in all cases. So these viruses doesn't kill the nucleus. I don't think the histology was showing nucleus damage and so on. That was a very gross view. Those were histology pictures taken a long time ago. I think the idea there was to show that the tissues are not necessarily damaged in the cases where there was clearance and persistence, but they were damaged in the case of immunopathology. Nirikshit asks, does exhaustion mean number of CD8 cells going down or their antigen clearing ability reducing? Yeah, the number of cells don't necessarily go down. Their antigen clearing ability comes down. Anonymous says, would low viremia patients still infect others? Yes. Amya Dravid asks, why do the circles peak and then recede for 2 weeks in the HCV data? That's because of the immune responses. They start controlling the viruses spontaneously. Mahesh asks, what would be the challenges in devising a dialysis system that would isolate immune cells and treat using these antibodies ex vivo? That's a tough question. There are people who do that. There are these studies called plasma apheresis studies where people filter out plasma, they remove viruses from circulation and bring them back. But the specific challenges involved there are something that I will not be able to answer. Amit Singh asks, HIV elite controllers are able to suppress HIV for a very long time. Are there any differences in CD4, CD8 dynamics in these patients? And can it be possible to use this information as a biomarker to figure out host susceptibility or severity to HIV infection? Amit, great question. Elite controllers are different from the post-treatment controllers. Elite controllers actually have a much more robust immune response. They're able to target a diverse collection of viruses upfront. So they rarely let the viruses, if they're not treated, flare up into progressive disease. Whereas the post-treatment controllers don't necessarily have that. And what these antibody treatments and other treatments are able to do is, with the existing immune response capabilities, they're able to drive the system to a state where those capabilities are enough to bring about lasting control. Now, biomarkers are difficult to identify. People are struggling with what would be the ideal biomarkers for such cases versus elite controllers. I think the answer is not settled yet.
There's a question, what is the minimum viral particles per milliliter for COVID to be infective?
I don't think people know that. The only thing they can say is the level at which you get symptoms versus you don't. And I think about 100 copies, and I'm not even sure you can call this per ml, because those are all based on swabs that people have collected. And so there's a limit of 100 copies that people say is where you get symptoms. Asymptomatic people can infect them, and not everybody who has high virus titers gets symptoms. So there are many asymptomatic people who have high virus titers also. I don't think there is a good answer to this question. People have made predictions about whether you would get significant infection after catching viruses to a certain level. So if you get an infection, if you get viruses into your body, then what is the amount of virus that you should get inside that causes the infection to sufficiently flare up? This estimate is there, but how that relates to the amount of virus that the transmitter carries is not something that is well understood.
So there's a question, even though IgG concentration may have been lowered across time, does the body have the capacity to ramp up production if re-exposed to the virus?
Yes, good question, and the answer is yes, because in general you have memory B cells which are also formed as a response, as a part of this germinal center reaction, and they then preferentially seed germinal centers when somebody is re-exposed to the infection, and that's how you improve the antibody responses. And partly that is what is going on also with the booster shot of the vaccine. Do serological surveys capture the entire population of different antibodies? I'm not sure about the surveys per se, but people have been able to look at the diversity of antibodies that are present in individuals. Most antibodies, so the whole focus of our talk was neutralizing antibodies. There are a lot of antibodies that are not neutralizing, which don't prevent the virus from causing infections. So you will find a lot of reports documenting a lot of those antibodies, but they are not of much consequence. The neutralizing antibodies are a lot fewer, and many studies have documented them. Is it practical to monitor the CTL response to devise a treatment plan? I would argue yes, but as far as SARS-CoV-2 is concerned, more data is needed to establish that, although growing evidence is suggesting that exhaustion, CTL responses are critical to final clearance of the infection. In other settings, absolutely. For HIV, I mean, there's a clear case that CTLs are important. Is it possible to customize a vaccine to an individual? Wow. That would be tough. In fact, at a very gross level, people are beginning to do that to strains of the virus. So for example, now we know all these mutant strains of the SARS-CoV-2 that are floating around. If one has to use the same vaccines, one has to establish that responses are similar. Even that is becoming hard. Tuning vaccines to individuals is really difficult. And also it's probably not practical unless somebody has some way of doing that in a scalable manner. Is it early for learning-based approaches for modeling the dynamics? No, people are using it, especially because of the diversity of the viruses, the diversity of the antibody responses, and the number of factors that is involved. There have been machine learning approaches that have been used, and even more so in thinking about fates of cells, where although I talked in a superficial manner perhaps about CD8 T cell exhaustion and activation and so on, those are actually deep states of the cells that are realized as a consequence of a large number of signaling events. And there too, the players that are involved are not well understood. And a lot of machine learning approaches are being used to deduce conditions that would give rise to one versus the other state. Dynamics, of course, can be then studied as well. Can previous exposure to related coronaviruses, which leads to antibody generation, circumvent the need for boosters? People are thinking about that much. So the 5% people that suffer mortality is just a gross number. It varies very much by region, by gender, by age, and so on. In India, for example, that number is much lower. And one thought is, although it's not proven yet, that we have been exposed in the past to circulating human coronaviruses that are much less lethal than SARS-CoV-2, and there possibly is cross-reactivity there of the antibodies and T cells, and because of which we might actually be better protected. Whether that will allow circumventing the booster is something that can be thought of depending on how much protection there is, but that has to be quantified. Usha asks, to predict how long immunity would last, one needs to know experimentally the quality of the antibodies, or are there ways or biomarkers? So at the moment, there are no, so I'm not going to say for everybody one needs to do an experiment, but right now, even at the first sort of cut level, good markers of how long these antibodies last are not there. So the Moderna studies are saying that you can go to up to 3-4 months. They have measured. I mean, that's how long the studies have been conducted. But going forward, there will be measurements of antibodies that people can make that will suggest the extent of protection. But this is I think, a more difficult question to answer because of memory B cells that might be there, which can then trigger antibody responses following reinfection. Also, CD8 T cells, which are not being looked at much in the current vaccine studies because initial studies are suggesting that they are not as important as the antibodies for effecting a cure. But going forward, they might actually be also something that needs to be looked at. At the moment, the first level of experiments have to be done just to document the level of antibody persistence after vaccination, based on which then biomarkers and other ways of estimating the duration of protection can be identified. Does your study suggest that any therapy which can boost CD8 can help tackling most forms of viral infections? I'm not sure that one can generalize it that way. It would help to have CD8 T cells boosted, but blanket boosting of CD8 T cells is a risky business because you can boost them non-specifically, and then they can then flare up and cause other kinds of autoimmune reactions. Specific boosting is not easy. People are trying that. And this is where I think partly all of these CAR T cell therapies and so on are being attempted. They're not boosting necessarily, but they're introducing special kinds of CD8 T cells that are going to be specific to the targets. Ramakrishnan asking, does clearance really mean complete removal, or could there be a low level of persistence that one could live with without having any symptoms? Again, a great question. As far as SARS-CoV-2 is concerned, we don't know whether clearance also means some lower level of the virus that we can live with. there have been complications that people have experienced many months after they have been declared cured of the infection. What the causes are not very well understood, and it could very well be that there is some low level of the virus below detection in some parts of the body that people have not measured or not looked at. With HIV, of course, the evidence is clear. You rarely get complete clearance of the virus, but in these post-treatment controllers, the virus is kept under check so that one can live then much longer than otherwise. Is immune cell exhaustion central to all infections and clearance mechanisms? Yes, there are very few. I mean, in fact, I am not sure there are cases reported where exhaustion never happens. It may be less or more depending on how much infection there is, but there is no reason for us to believe that there might be a case where no exhaustion will take place. Deepak asks, if there are shared epitopes with infections like polio, MTB, how will the immune response manifest over time? The shared epitopes, you would actually mount the response much faster. And therefore, if that helps one get rid of the infection faster, the response would also fade away faster. You would peak faster and fade away faster. That would be my guess. And Narmada asked, this is the last question, do any of the HIV analyses depend on the mode of transmission? I recently came to know that pre-exposure prophylaxis has different efficacy for these two modes of transmission. Why is that? It's a great question. We haven't, in this study, talked about the modes of transmission. We have independent analyses where the mode of transmission does make a difference. And the reason it makes a difference is because the barrier that that the virus has to surmount to cause systemic infection is different across the two barriers. And that's the reason pre-exposure prophylaxis also works differently. The thought is that men who have sex with men have barriers that are much easier for the virus to cross, and therefore the thought is that less fit viruses can get across, and whether they cause less severe infection or not is something that is yet to be established. Navkant, I think that's it. I've looked at all the questions here.
Right. Yeah. Excellent, excellent. Thank you again, Narendra, for the fantastic presentation. And I would also like to thank all the audience who participated very actively. I wish this would have been a personal interaction. And I would also like to thank Mr. Veeranna and his team for enabling all the IT support. Thank you again, and we'll close the session now.
Thank you very much.
Thank you.
Thank you, sir. Thank you, everyone.
Thank you, Veeranna. Thank you very much.
We are closing the sessions. Thank you.
Thank you. Bye.
Bye.